The first argument is intelligent design answers everything and therefore nothing. If you have an answer to a question that actually explains absolutely everything that ever happens, and that's the answer, how useful is that answer, basically? So that means intelligent design is utterly boring. If it answers everything, you know, what's the need of science? What's the need of research? All you say, well, I don't understand how that happened because God probably did it. No, I don't understand that, so God did it. Everything you don't understand, God did it. So that's kind of a boring answer. It doesn't really stimulate intellectual inquiry. And so that's a common argument against the whole, well, I don't understand, so God did it argument. The other common argument is intelligent design is a thinly disguised creationism. You know, creationism politically hasn't worked because the U.S. government is supposed to be freedom of religion, right? Not promoting any particular religion over any other. In our public school systems, it's not supposed to be promoting a particular religious view. And so they're saying, well, because creationism can't be taught in the public school systems, creationists have just wrapped it up and called it a different name, but it's still the same thing. They've just called it intelligent design theory, but it's really only creationism in another disguise. That's the other argument. Third argument is that intelligent design uses God of the gaps arguments, which is supposed to be a logical fallacy, a fallacious argument. The gaps argument is a common false argument, a style of argument. A fourth argument is that intelligent design proposes no testable, falsifiable predictions that have not already been falsified. So even if intelligent design might have a few points, or might used to have a few points, the few points that may have had once upon a time have been falsified, they've been disproven. For example, irreducible complexity proposed by Beatty in 1996, that's been disproven, and specified complexity proposed by Dembski, a mathematician, that's also been disproven. Those things don't actually exist anymore. The fifth argument is that no intelligent God would have done it that way. There's all these mistakes in nature, you know, low back pain, people die, people get sick, animals aren't, you know, your eyes wired backward, your jaw's not the right size. All these mistakes, anybody with half a brain, they wouldn't have done it that way. I could have designed things better than whoever designed the natural world. So those are the arguments I hear commonly. And here's some of the answers, at least for me, that make sense to me that I've come up with to answer these rebuttals.
Can I say something?
Sure.
How can the human body, as complex as it is just the eye itself, be an actual substance?
Well, that's a common creationist point. A gentleman asked, how can something really complex like the human body have just evolved, just happened? Well, it's really, you know, I used to laugh at that too, in grade school and high school, and say, it's obvious things are designed, you know, right? So obvious. So how could people that are really intelligent like professors or mainstream scientists that are brilliant men and women, actually think that it wasn't designed? Richard Dawkins wrote a book entitled Climbing Mount Improbable. And the reason why they believe this is because they don't think it happened all at once in one big bang or an explosion producing a Boeing 747 or something. They think it happened in little baby steps over millions and millions of years. And if you look at that argument superficially, even in fair depth actually, it makes a lot of sense. That's why this argument, this whole topic is not an easy topic. If you really look at it carefully, the Darwinian perspective makes a lot of sense. And that's why it convinces a lot of people. You have to actually, it took me about at least 4 years to figure out why it didn't make sense. So, and hopefully we'll get into that a little bit today in particular. The everything and therefore nothing argument that explains everything and therefore nothing. Let me ask you, are hypotheses that explain absolutely everything, can they be useful? You know, is it, like for example, if you had somebody who was super intelligent, had all the power that you would ever need, would somebody like that be able to explain everything? Just about, you know, everything physical anyway that you could think of. Even I can explain a lot of things with my own creative powers. I can make a lot of stuff. Human intelligence can make a lot of stuff. We can explain a lot of things with just human level intelligence. But that concept, to me, is actually quite useful, because it's useful in the sense that not everything can explain everything. For example, there are some creative forces, like a tornado. Tornado is a force of nature, right? Can it explain everything that humans can explain, that human intelligence can explain? No, it's limited, right? So if you see something like a granite cube, I just love the granite cube. If you see something like a highly symmetrical, polished granite cube, or the Statue of David, or Mount Rushmore, or something like that, and someone says, you know, a tornado did that. Does that make sense? No, because we know tornadoes are limited in their creative ability. But what if someone says, well, some intelligence did that? Does that make sense? Right? Because intelligence can explain more. So the bigger the intelligence, the more you can explain. So is that concept actually meaningless or not useful? Something that can explain everything is actually quite a useful concept, because it can distinguish between things that can't explain everything and things that can. You see what I'm saying? And beyond that, it's a logical fallacy for the evolutionists to accuse us of saying, look, your God can explain everything and therefore nothing. Because look, on the flip side of the coin, they say nature can explain everything, right? So what happened here? You don't understand the Big Bang, right? How did the Big Bang come about? How did the first cell come about? Well, we don't know, but we're sure nature did it, right? They make exactly the same argument we do. And ultimately, do we know how God made everything? No. Ultimately, we both come to a brick wall where we say we don't know. If we knew everything, or if I knew everything, how God did everything, I would be God, right? So, and if they knew everything that nature did, or whatever, they would be as powerful as nature, too. So it's really a false argument, as I see it, from their side, for several reasons. Also, beyond that, they think that they can detect intelligence, very high level of intelligence, even higher level than human intelligence. They just call it extraterrestrial intelligence instead of God. What about the intelligent design theory is boring? You know, here is William Provine. He's a science historian at Cornell University. He's fairly well known in scientific circles. He writes, the most basic problem with intelligent design is that it's utterly boring. Everything that's complicated or interesting about biology has a very simple explanation. God did it, or intelligent design did it. The thing about this is that SETI scientists are looking for particular types of radio signals coming from outer space, that they would say, if they found the type of signal they're looking for, they would say, aha, this is evidence of intelligence in this radio signal. Now, if such a signal were ever found, would anybody consider that boring? No. That would be exciting, right? Would it hit the front page of every newspaper in the world? Yeah. Right. So, I mean, that's not boring. Computers also have a very simple explanation, like humans did it. Right? You wouldn't say all the moving sands in the Sahara Desert over millions of years came up with an IBM. That's not a reasonable explanation. It's better to say some intelligent force did this. And it doesn't make it simplistic or boring just because you say, I don't believe the blowing sands in the desert and some lightning strikes did this. That's not boring, it's actually interesting. Because then you can move out of a realm of study, of just determining why certain forces can't do things and why other forces can. And that's very interesting. Also, if you just want to get matter of fact about it, sometimes simple truths are maybe boring, but that doesn't make them false. Like 2 + 2 = 4. That's kind of mundane, boring, but is it false?
Yeah.
It's true. I mean, 2 + 2 = 5, that's more interesting, right? But is it true?
Yeah.
Okay. What about intelligent design as a religion, therefore it's not a science? The argument is that religion talks about non-physical, non-testable, non-falsifiable truths, where a scientist talks about the hard, concrete, something you can see and feel and touch. So, is that a valid argument? Are there any examples of non-falsifiable truths that you actually believe that you can't test, that you just inherently know? For example, love. Can you test love? Or do you just know? Like, for example, I love my wife, Sigrid. Do I need to test that? Or is that falsifiable? Is there any experiment I can set up to say that do I love her or not? No, not for me. I know it internally. How about my hypothesis, though, that Sigrid loves me? Is that testable or falsifiable? For example, let's say she's I come home one day and she hit me on the head with a frying pan. And then she starts yelling at me, and she does that every day for a year. Would I begin to question my love hypothesis? You know? So there's different things. Some people say love is not testable. Well, in some sense, that's true. My love is not testable because I know it internally. I don't have to ask somebody else if it's true or not. However, but somebody else, if I believe somebody else loves me like God, does God love me?
Yes.
That's testable. That's falsifiable. There should be evidence for that. Otherwise, if God started beating me up every day and doing bad things to me, and there's no evidence I could see anywhere where he could show any love for me that I could actually see, feel, and touch at all, then I might be questioning my love hypothesis about God. What about, the same thing's true for joy, beauty, taste, desire. I like vanilla ice cream. Is that falsifiable? Testable or anything? No, I can't even explain it. I just like vanilla ice cream. You know, there's no argument. No one can challenge me. No, you don't. You know, I do. No one can argue. The same thing, well, actually mathematics used to be thought to be that way. Recently, though, in the last 10 years, I would say, mathematics has actually become more of a testable science, even falsifiable. There are many little subtleties in mathematics these days that are not so clear-cut, that it's not any absolute anymore, which is very interesting, I think. There's actually certain things mathematicians believe that aren't even actually they're not provable at all. They're not absolutely provable. They just believe them based on the preponderance of the mathematical evidence. And we can get into that more later if we don't have time today. And what about God? That's what this is all about. Is God falsifiable, testable, or is he just some feeling I know inside of me and that's it? Is God like vanilla ice cream? Okay? Or does he have some sort of external, physical, testable property that he reveals himself outside of my own internal revelation? That's the question. Intelligent design uses God of the gaps arguments. This is a challenge. The problem with this fallacy, people always say, oh, the God of the gaps fallacy. To me, it's not a fallacy because all scientific hypotheses are based on the gaps argument. No hypothesis in science is 100% provable. The scientific method only narrows the place to where truth can be found. It's able to disprove things, but it can't really prove anything. It only is able to say that things aren't true. It can't say that things are absolutely true. So what scientific method does is it narrows the gap. But it's still based on the gap argument. After a point, you have to take a leap of faith and jump across the gap. Because nothing is absolute, even in science. The argument is, well, you know, you may have had some points once upon a time with intelligent design theory, but all of those arguments have been falsified. What's interesting about that argument that I hear all the time is that that argument in itself means that intelligent design theory used to be a valid science, which no one really says in scientific literature. Everybody argues that intelligent design theory is a religion. It's not a science. But if you make the argument that, well, some parts of it have been falsified, when you make that argument, as soon as you do that, you qualify it as a science, at least in the past. Because if you have a theory that's open to testability and falsification, even potential falsification, you have basically said it's a science. It's a valid scientific endeavor. In particular, though, let's look at what they claim has been falsified. Irreducible complex systems do not exist, and random mutations combined with natural selection easily produce Dempsey's complex specified information, and we'll get into what that CSI means in a minute. There are no irreducibly complex systems. Here is Kenneth Miller. Here's a picture of him. He's one of the most famous apologists for against intelligent design movement in the world today. He's from biologist from Brown University. He's written several well-written books. He's a very entertaining speaker and a brilliant man. He writes, the logic of their argument, intelligent designists, is that you have these multi-part systems and that the parts within them are useless on their own. The instant that I or anybody else finds a subset of parts that has a function, that argument is destroyed. For example, let's take a car. And you take the motor out of the car, does the subset of parts, any subset of parts in the car system still work? Yeah, like you can still turn on the radio, right? And the lights still work probably, at least for a while, right? The same thing is true with fish that have lost their eyes, like cave fish. You take the eyes out of a cave fish, does the rest of the fish still work? Right? So intelligent is that fish therefore not irreducibly complex? Because you can take the parts out of it and the rest of it will still work. So therefore the fish is not irreducibly complex, according to Kenneth Miller. And it seems to make sense. And we'll get into a little bit of a clip from one of his lectures to explain it from his own words a bit more specifically in a minute. This is Steph Ryder. He doesn't look like much, but he's a smart guy. Kurthan, Steph Ryder for Life Science. All of the systems that Beatty claims to be irreducibly complex really aren't. A subset of bacterial flagellar proteins, for example, are used by other bacteria to inject toxins into other cells. So you can take out parts of this flagellum, and it still works as with its own function to inject toxins. It's a toxin injector now instead of a flagellum. And here's the flagellum. It looks like a machine. Some people have said it looks like an outboard motor. It's got a drive shaft, rod, stator, rotor, propeller, filament, a hook, bushings. It's got all the pieces and parts, the basic parts for an outboard motor. So you would say complexity, well, this is very complex. And this gentleman's argument would say, how in the world can something that complex with all those parts just evolve? Well, it's really actually quite logical, according to Kenneth Miller.
One machine particularly attracted his attention.
I'm sorry, before we get into this video, this is Michael Beatty and his initial approach to the flagellum and why he thought it was designed, and his initial ideas behind irreducible complexity.
One machine particularly attracted his attention.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called the bacterial flagellum with all of its parts and all of its glory. It's had a propeller and a hook region and the drive shaft and the motor and so on. I looked at that and I said, that's an outboard motor. That's designed. That's no chance assemblage of parts.
Beatty's reaction was not surprising, for the molecular motors that drive bacteria through liquid each depend upon a system of intricately arranged mechanical parts. These parts come into focus when portions of a cell are magnified 50,000 times. Biochemists have used electron micrographs like this one to identify the parts and three-dimensional structure of the flagellar motor. In the process, they have revealed a marvel of engineering on a miniaturized scale.
Howard Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
And even though they're spinning that fast, they can stop on a dime. It only takes a quarter turn for them to stop and shift directions and start spinning 100,000 RPM in the other direction.
And just like outboard motors on motorboats, it has a large number of parts which are necessary for the motor to work.
The bacterial flagellum, 2 gears, forward, reverse, water-cooled, proton-motive force. It has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller, and they function as these parts of the machine. It's not convenient that we give them these names. That's truly their function.
Okay, so that's the argument. Now, what does Miller say to counter that? How does he explain how that all happened? I mean, it seems obvious, right? This had to be designed. It's an outboard motor, but not so quick.
The notion that these complicated biochemical structures couldn't have been produced by evolution has been championed by Michael Beatty. And Beatty has an idea that he calls irreducible complexity. And he says, you can't evolve these things because they're irreducibly complex. Notice what he says. An irreducibly complex system can't be produced the way that evolution works by numerous successive slight modifications of a precursor system, because any precursor to an irreducibly complex system that is missing a part is by definition non-functional. These are multi-part systems. And he's basically telling you that the 30 or 40 proteins that are in here, they all have to be together and there's no function. And since natural selection does have to work gradually, I agree on that point, it can't produce 20, 25, 26 proteins knowing what will eventually happen because natural selection is blind, which is indeed absolutely true. So the poster child for intelligent design by any standard, it shows up so often, it really could be called the poster child, is in fact the bacterial flagellum. This was mentioned so often in the trial that the judge, probably from fatigue, got a little sarcastic about it. One of the attorneys said, "Your Honor, when we reconvene, we're going to talk again about the bacterial flagellum." And the judge at one point said, "Oh, goody." The last expert witness for the Board of Education, a biochemist named Scott Minnick from the University of Idaho, was called up to the stands to talk about this. And since Behe had talked about it, the lawyers had talked about it, they had argued about it, and I had talked about it, as I'm going to show you here. For a second, Minnick got up there, and he said he was going to talk about the bacterial flagellum, and the judge deadpanned, "Well, we've heard that before." And Minnick turned to him, this is the best line of the trial, Minnick turned to him and said, "You know, Your Honor, I sort of feel like Zsa Zsa Gabor's fifth husband. I know what to do, I just don't know how to make it exciting." So I take my hat off to Scott. That was good. I like it. So what is this argument about? Here's the argument in very simplified form. If you have a complex, multi-part biochemical machine composed of many parts, its function, everyone agrees, can be favored by natural selection. But the argument is that evolution can't produce them because the individual parts have no function of their own. That's what irreducible complexity means. So natural selection can't make this, doesn't have any function. Can't make that, can't make that. Therefore, you can't evolve a structure like this. Now, how does evolution explain something like that? Well, ever since Darwin, we've had a very good explanation. And that is these complicated machines, they don't arise from scratch. They arise from combinations of components that have different functions of their own. And the components originate with functions of their own as well. Therefore, natural selection will work every step of the way. Now, that's not evidence, that's just an argument. But the beauty of this is we can now hold these two ideas up against each other. And we can say, who's right? If irreducible complexity is right, then the parts of these machines should be absolutely useless. But if evolution is right, we should be able to take these machines, look at their parts, and discover, wow, they do other jobs. So let's go ahead and do that. Let's take the bacterial flagellum. So if we start with the flagellum, here it is, and these drawings name the genes and the proteins in the flagellum, and we say, let's take away a whole bunch of the parts. How many? Not 1, not 5, not 10. Let's take 40 of its 50 parts away. Now watch very carefully, because I'm going to do that experiment right there. There it goes. The parts are all gone, and I have left 10 parts that span the membrane. What are left behind are 10 proteins in the base of the flagellum. Now, if irreducible complexity is right, this should be absolutely functionless. It should have no function. But if you will pardon the double negative, what is left behind is not non-functional. What is left behind is the type 3 secretory system, and it is fully functional. I know most of you in the room are going, of course, the type 3 secretory system. The type 3 secretory system is a molecular syringe in which some of the nastiest bacteria on this planet produce toxic proteins, grab onto one of our cells, and inject those proteins into our cells. The bacterium that causes bubonic plague works this way. It's really nasty stuff. Well, guess what? The 10 proteins that make up the type 3 secretory system are directly homologous to the 10 proteins in the base of the bacterial flagellum. They don't produce movement. They're not a flagellum. But are they functional? They are fully functional. So remember that claim. Any precursor to an irreducibly complex system that is missing a part is by definition non-functional. This guy is missing 40 parts, and it is perfectly functional. What that means, there's no other word for it, is that that statement is wrong. Now, that's not an incidental statement. That is the heart and soul of the intelligent design argument. And in this case, it turns out to be wrong. Now, it's even wronger than that, because it turns out that not only do these proteins make up the type 3 secretory apparatus, but almost every protein in the bacterial flagellum is strongly homologous to proteins that have other functions elsewhere in the cell. And what that means is when we look at this wonderful icon of intelligent design, a careful analysis of the flagellum actually matches evolutionary theory, namely the parts should have functions of their own and not the intelligent design prediction. And that's simply a fact.
Case closed, right? All right, see, he just falsified intelligent design theory in one fell swoop in 5 minutes, right? That seems pretty convincing. I mean, if you just sat in the audience and you didn't know anything else besides what he just told you, he makes plenty of sense, doesn't he? To me, he does, to be honest. All right, so let's look at this a little more closely. Now, here's a picture here of the type 3 secretory system that he mentioned, that toxin injector, bubonic plague and all that. That's real. It's a real toxin injector. It's like a little syringe, and it injects toxins from bacteria into mammalian cells, and it kills them. Here's the flagellum. You chop off this part of it, and you're left with the toxin injector. Now, it doesn't move. It's not modal, but it does work. He's right. Now, which of these things, according to evolutionary theory, according to Miller's argument, would you think logically should have evolved first? This one or this one? This one, right? It's simpler, right? It's only got 10 of the 50 proteins needed, right? Is that true? Okay, so let's look at the toxin injector system a minute. The flagellum is found in many kinds of bacteria, gram-positive, gram-negative, pathogenic, non-pathogenic, all over the place. Toxin injectors are restricted to a few gram-negative bacteria that attack only plants and animals, which are thought to have come along billions of years after the flagellum is thought to have evolved. Kind of interesting, huh? Okay, there's little similarity or homology to anything within less complex motility systems. There's only homology to the flagellum subset within the toxin injector system. That homology means similarity. Okay, so just a fancy word. Several scientists recently, in the last 8 years, like Nugent and Paulson and several other guys I can't pronounce their names, but they have said that the TTSS system, the toxin injector, evolved from the flagellum, not the other way around. It's a degenerative change. In other words, you chop out the fish's eyes. Is that easy to do? It's simple. You can do it with one point mutation. Okay? How easy it is to chop something. How easy it is to break Humpty Dumpty. It's easy, right? It's easy to break something. Why? Because there's so many ways to break something. You break it, you can drop a glass vase. Is it going to break the same way every time? No, break a bunch of different. There's so many ways to break something. So it's easy to break something. It's easy to take away parts from a system and still have subparts and subsystems still work. That's why it's easy to shoot a gun into a car and go right through the engine block, and the engine won't work, the car won't run, but the lights may still work for a while. That's easy to do. And so, now what about putting Humpty Dumpty back together again? Is that so easy? What if you never had eyes to begin with? The fish never had eyes to begin with in its ancestry for like 10,000 generations. What are the odds that it's going to get eyes back with a single point mutation? Not very good, right? You've got a very complex system. Well, the same thing for this bacterial flagellum. What's interesting about his argument is he deleted 40 of the 50 parts. What happens if you only delete one part? What are the odds that the 49 parts will be functional? First of all, you won't have a functional flagellum as a motility system, and you won't have a decent toxin injector either. You have to delete a whole bunch. Now you're down to the toxin injector. How are you going to get the motility and function back again? Is that going to be so easy? It's very easy to go downhill. It's a whole other thing to go back uphill. And we're going to talk about the complexities of going uphill versus downhill. A bit about the mechanism. Random mutations, as we talked about yesterday, they're real. They affect DNA, the coded molecule in our bodies that make us who we are. And they must be inheritable to affect evolution, or to be affected by natural selection. Natural selection is also a real force of nature. It's a mindless process. It has no goal in mind. It doesn't care about you or anything else. It is capable of detecting phenotypic changes, which basically means what you see and feel and touch. It can't affect the genotype, which is the coded information. It can't see that at all. It can only affect what the genotype makes, which is called the phenotype. This is a schematic drawing of DNA. It's made up of 4 characters, A, T, C, and G. It's kind of like computer codes. Instead of 2 characters, computers have what, 0 and 1 for their code? DNA is more refined. Computer engineers have marveled, wish, in fact, some computers are even based on DNA nowadays. But it's based on 4 characters. So you can make a higher compression of information using DNA than you can in computer code. However, it's similar to all other coded languages, like Morse code, English, computer code, all other language systems, because it's a symbolic code. The order of the symbols are meaningful. Does that make sense? There's also, you have to get the information out of DNA. So once it's there, how do you get it out to do something with it? Well, there's these fancy words called transcription and translation. Transcription is where you copy the information in DNA itself into a working copy called RNA, messenger RNA. And that's called transcription, how you make that messenger RNA copy. It's transcribed. Okay? Then, once you have the messenger RNA, you translate it into a completely different language. Messenger RNA is almost the same language as DNA, just one little minor variation. But translation into proteins, that's going from one language to a completely different language. Proteins are based on 20 characters instead of 4. There's 20 different amino acids for the characters for protein building. And so you have to translate from the 4 characters to the 20 characters. Does that make sense? Like going from Morse code to English. You have to do a bit of translation, right? So to decode the DNA is coded in triplets, 3 letters for a word. And then that 3 letters makes a protein called serine. That's the code. It's arbitrary. It didn't have to be that way. It's arbitrarily set up that way. And then the proteins, of course, are folded into tertiary structures, 3-dimensional structures, beyond the 2-dimension of the code itself. And it's actually the 3-dimensional structures that are functional in protein shapes.
Translation is the process by which the...
Let me see if I can use the sound.
Translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has 2 binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome. A single messenger RNA molecule can be translated a number of times and produce many molecules of polypeptide.
Okay, it gets more complicated than that, but that's complicated. A lot more complicated than that, but that's as complicated as we need it for the purposes of this discussion. And just that in itself, you have to have all that system in place before you can have the first cell, the first living cell. And there is no natural selection before you get all that in place. But beyond that, let's just give them that. You have the first cell. Let's go from there. Okay. Proteins. Just to get it, you know, this whole process, the goal is to make proteins. Proteins are the functional units of the cell. They're very specific in their sequential order in order to have the proper function. There is some flexibility, like in a paragraph. Can I change a couple letters in a paragraph and you pretty much still read it? Same thing with proteins. You can change a couple of letters. Most of the letters can change at least somewhat. But still, overall, can I change half the letters in a paragraph and still make sense? Probably not. And 90% of the letters in a paragraph? No, almost certainly not for most paragraphs. And same thing for most proteins. And some proteins are more specific than others. What if I write a really technical paragraph for NASA, how to put O-rings in the space shuttle? Can I change that much at all? Probably. No. So there are some really specific proteins as well that you can't change much at all. And so it all depends on the job of the protein, how flexible it is. And so there's some sequence requirements there for every protein. And so in that particular sense, all proteins, even single proteins, even little proteins, are irreducibly complex. Because if you reduce them a certain amount, they no longer do the particular function for which they originally had the ability to do. Same thing for the flagellum. You already take out the 40 amino acids, yeah, it still has the toxin injector function, but does it have the flagellar motility function? No, so the flagellar motility function is irreducibly complex. Because you take out one of those 50, you lose that, boom, gone. Same thing for the toxin injector. Toxin injector function requires 10 proteins. You take one of those, you go down to 9, you got toxin injector function anymore? No. But do you still have single amino acid protein function? Yeah, you still have. You can go all the way down to single atoms if you want to make that argument and still have functions left. But you don't have the function in question. Every functional ability has a minimum part and specificity requirement for both size and sequence. Does that make sense? Okay, I'm going to take a little side step here. This is a little bit off topic, but it kind of plays back into it later on. So hopefully we won't get lost here. I want to talk a little bit about Mendelian variation, because a lot of people think, even Darwin thought, Mendelian variation is evolution in action, and it's not. Mendelian variation is a completely different form of change that's really not the production of anything new when it comes to the gene pool, or genetic functions, or genetic information. For example, all these finch peaks, the finch heads and finch peaks, they're all different. And that's what Darwin noted on the Galapagos. Same thing with the peppered moths, some are light and some are dark. Depending on the environment, you can select for one type or the other. Same thing with all these breeds of dogs. Very different looking dogs, all came from the same gene pool. Is that evolution or not? Did anything new come into that gene pool to produce all those varieties or not? Well, the answer is no. Gregor Mendel, first, he's the father of genetics. He actually lived in the same time during Darwin, but they didn't talk to each other. They didn't know about each other, unfortunately. Because if Mendel had become famous first, Darwin would have had a much harder time presenting his theory of natural selection, random mutation, natural selection. Natural selection does play a part in Mendelian change, but the random mutation part isn't really random. There are random aspects to Mendelian variation, but there are also very strict rules to Mendelian variation. For example, chromosomal crossover. You take the chromosome from your mother and your father, and they randomly cross over, and then you take pieces and parts from each of your mother and father's chromosome, and you make the kids. And that's why no one's brother, and also the crossovers are not in the same spot each time. They can cross over at different spots and make a different genetic complement for the children. And that's why me and my brother look very different. He's dark and fuzzy, and I'm white and have no hair. So we look very different, but we both came from the same gene pool. Is it because we were mutated versions of our parents? Well, sort of, if you want to use a loose definition of mutation. But it's a very specific mutation. It's a very specific crossover none of the genetic elements from our parents were actually distorted in any way. We just happened to get different portions from our parents that still ended up in the same location on our individual chromosomes. Our actual genes, the discrete packets of information, were not altered between me and my brother. And in other words, we are different expressions of the same genetic pool of options that my parents had access to. They have a very large pool that includes trillions and trillions of potential options for offspring, and me and my brother just happened to end up in the 2 spots that we ended up in. But that is not the same thing as producing new information. I had no abilities, genetic abilities, that my parents did not have in their gene pool. We basically talked about this pretty much, except for illustration. Let's say I have a bicycle, a blueprint for a bicycle. And for the handlebars, I have those big loop handlebars for the beach bikes, you know, and then I have the little racing ones that are curved over for the 10-speed bike. And let's say I've had 4 or 5 handlebars options for building my bike. That's a locus, a genetic loci for the handlebar options. What happens though if I mutated my blueprint so I put the handlebars where the pedals are supposed to be? Would my bike work very good? Or what if I put the pedals where the wheels are supposed to be? Would my bike work very good? No, but that's evolution. That's actually a true mutation that produces something new as far as a functional difference. Whereas before, I could just say, as long as I put the different options that I have in the right place, that's Mendelian variation. You see the difference? Okay, we talked about that. Gregor Mendel, he just came up with these different traits. Flower color, purple or white. These are all gene pool options. But, you know, you can get a white flower with an axial stem that's short, round, yellow, constricted, and green for a pod color. You know, you can do a whole bunch of different variations. You can get tons of different kinds, but it's all Mendelian. None of this is anything other than a P. Okay. Oops, sorry. Okay, dominance and recessiveness. So how do you know if you're going to be hairy or no hair at all like me? Like my brother or hair like? Well, it depends on which is dominant and recessive, and there's also co-inheritance, and it gets much more complicated. But it doesn't mean that you don't have the genetic features that were already in your gene pool necessarily. It means that some are expressed and some aren't. For example, let's say the parents had the green and the yellow option, and this is a dominance and recessive. And so what are the second offspring going to be all yellow if the yellow option is dominant? They're going to be all yellow, even though in their genotype they have the genes for both yellow and green. You understand? That they're all going to be yellow even though they have the genes for green. But in the next generation, they might have a green child 25% of the time, because recessiveness is all you have is just recessive genes. You have no option but to be green. Make sense? And so that's how you get all these variations in Mendelian variation in a nutshell. But it's still a static gene pool. The gene pool itself has not achieved any further options. You still have the same 5 different options for your handlebars. You haven't produced a different form of handlebar or a different kind of wheel option. So in order to get more options, you have to actually have mutations that mutate the genes that are already there. Besides, or else your gene pool is just going to stay with the static gene pool. You're not going to turn a dog into a cat or a lizard into a bird. So, evolving gene pool, you have to make more stuff, more options. So let's look at the odds of that. In order to get more options, it's kind of like getting different kinds of words that you don't already have in your dictionary. Okay? So how hard would that be to get a new word that's not actually in your dictionary? It may exist out there in the universal dictionary, but you don't know what the universal dictionary says. You only have access to your little tiny little dictionary. And so how do you add from the universal dictionary to your dictionary? The only way to do that is by random mutation natural selection, real evolutionary events. And so in order to search in the huge dictionary out there and the potential of all of sequence space, you have to look randomly through it. You don't know where anything exists in that dictionary, so you have to do a random search. And that's where random mutations come in. So let's say I need a new 3-letter word. What are the odds that I get another one from out there in the ether that I don't yet have? What are the odds that I come across a new defined 3-letter word that's beneficial for me in my current environment? Well, you have to look at this. I look at it, how many total potential three-letter words are there? Well, there's 17,576. That's an absolute number. You just take the number of characters that you have, it's 26 plus space for 27 characters. Add that to the power of 3, and the answer is 17,576. That's your checkerboard. That's your sequence space of options. So you have 17,000 squares that you have to search through randomly. Well, what are the odds that you'll come across a square that actually means something? Well, that depends on how many three-letter words are defined in the universal dictionary out there. Well, and let's just take it for an English language system as an experimental basis to look at this problem. In the English language system, the checkerboard that's an absolute size, there's no changing that. For two-letter checkerboard size, there's 677 options for that. Defined three-letter words in the English language system is 972. So that produces a ratio of defined versus not defined of 1 in 18. Does that make sense? So the odds of randomly stepping across on your checkerboard and landing on a square that means something is what? 1 in 18. So you have to take 18 steps randomly on average to hit on something. Well, that's not so bad. How long does it take to take 18 steps? 18 random mutations. Not very long. If you have a big population of 100 million people or 100 million bacteria or whatever, it'll happen bunches of times in one generation, right? Same thing for two-letter words. And then you can get from cat to dog, like yesterday, really quickly. No problem. What about seven-letter words? Your checkerboard size has now gone up quite a bit, right? Your checkerboard size is not just 17,000, it's 8 billion, right? So now, what's the ratio? You got 23,000 defined words. The ratio is now around 1 in 300,000. Now, if you want to include seven-letter sequences that include like a three-letter word plus a four-letter word or something like that, you might go down to 1 in 250,000. But that's still an exponential decline in ratio. You see what I'm saying? So the odds that you'll hit on something by a random walk is exponentially less. And so your random walk time is exponentially longer, even for the same size of population. Now, some people always say, well, you have a sizable population that's 100 million individuals like bacteria. You're still going to have every generation hit on one of these, but you're going to have exponentially less individuals hit on one of those. So the population that's successful is still going to be exponentially less, even though you'll be successful in each generation at this level. You keep going up levels, though, and that exponential decline, pretty soon you'll have no individuals in a given generation be successful. And pretty soon it'll take exponential amounts of time for anyone in a set size of gene pool to be successful at higher and higher and higher levels. That's an exponential decline in evolvability. For example, here's a short phrase like methinks it's like a weasel. It's famous. Richard Dawkins uses it in his methinks it's like a weasel illustration. The checkerboard size here is 27 to the power of 28. That's 28 zeros after that 27 number. It's a huge number. Just to illustrate the size of this, if each checker on our checkerboard was 2 centimeters across, there would be 1,000 trillion kilometers across. That's the size of our checkerboard. And there's only 93 million miles to the sun and 25 trillion miles to the nearest star. So that's a big checkerboard just for little checker squares, right? So how many meaningful 28-character phrases are there in the English language system? I don't know exactly, but let's say there's a trillion for illustration purposes. Well, if there were a trillion, then each of them would be surrounded by a vast ocean of 10 billion trillion meaningless sequences, on average. Some might be closer together because phrases and words and stuff in most language systems tend to be related to each other to make sense. But on average, there would be a fair gap between an average meaningful sequence and the next one. So changing 1 letter per second with no repeats, how long would it take? It would take 300 million trillion years to come across just one of the trillion meaningful sequences in our checkerboard with a decent-sized population of over 1,000 trillion. Let's say a trillion in our population. So what about just increasing the population size? If you increase the population size and you have more individuals looking and you can evolve faster. Well, that works. That's a great solution to the problem, except the environment can only hold so many individuals. Well, I got a limited little planet here, and then your little prebiotic soup, it's only so big, And even if the entire world were full of bacteria, it still can involve the bacterial flagellum this side of a trillion years. Not even one step in the evolution of the bacterial flagellum, if you do the statistics of it. So here's an illustration. Right next to you, this represents 3-letter words, and the green dot represents beneficial ones. It's easy when they're 3-letter words to get from one to the next, because they're so close together. When you get farther and farther apart, and then the meaningful sequences, the meaningful squares get spread out. It's not so easy to get from one to the next. That's sort of like stepping stones. Yeah, you can, in the bacterial flagellum, there's lots of potential stepping stones that are beneficial in the building or evolution of the bacterial flagellum. It's just that when you start at the very lowest level of minimum complexity in the flagellum, yeah, the next stepping stone is pretty close. It's easy to hop from that one to that one to that one without getting wet. But after you go up higher and higher complexity toward the flagellar motility function, you're starting to get upwards of 10,000 required codons of DNA, of genetic information. And the stepping stones at that higher and higher level get farther and farther apart. Pretty soon, you have to swim for it. And when you swim, nature is out of the picture. You have to swim randomly, your eyes closed. Good luck finding the next stepping stone. It's the same thing with illustration, a two-dimensional thing. At low levels, the land is very close together, kind of like chewing gum. You have nice bridges, and the ocean is pretty small. So you can walk around anywhere pretty much at will at low levels. You get higher and higher levels, the land starts getting stretched out like chewing gum. It has some bridges, but the bridges begin to snap at higher and higher levels, and pretty soon you get isolated islands once you get beyond 1,000 amino acids at minimum. And then even higher than that, flagellar level, you're really far apart in sequence space. I mean, universes apart in sequence space. And so getting from here to here is not a direct shot because natural selection is out of the picture. Nature can't see the genotype, remember? The genetic mutations, nature cannot tell that. What's the difference between Queasily Gook and Queasily Guck? Functional. There's no difference. So is nature gonna, if nature decides from one to the other to pass on the genetic information, Queasily Gook versus Queasily Guck, what's it gonna do? Nature's gonna say, oh, I don't know. He's gonna throw up his hands, flip a coin and say, Queasily Gook. There's no selective advantage from one or the other because they are both equally meaningless. They both have the same equal non-function. And so when you're in this neutral ocean or non-beneficial ocean, everything is equal. And so you do a random walk, and you do it all, and finally maybe you get to there eventually. But that's an exponential distance versus this linear distance. The longer this yellow line gets, the exponentially longer the red line gets, the actual random walk path. And it's actually more than 2 dimensions. It's hyperdimensional sequence space, which I really won't get into, but trust me, it's hundreds of thousands of dimensions. It depends on the size of the genetic sequences, how many dimensions you're dealing with. So Richard Dawkins to the rescue. Richard Dawkins says, oh, this is no problem. You can start with a random sequence like Queasily Gook or whatever, whatever that says. You start with that sequence that's 28 characters long. You take that sequence and you produce many copies of it, or offspring, right, in each generation. And you select in each generation those offspring that are the closest match to me-thinks it's like a weasel with your computer. Okay? How many generations doing that would it take to get from that to that? Me thinks it's like a weasel from Shakespeare. Only 40, 38, 41, an average of 40 generations. If you program your computer to do that. And he says, aha, evolution is easy. All you have to do is add a little bit of natural selection into it. What's the problem with this argument? There's no natural selection. What's the difference between blah, blah, and blah, blah? There's no functional difference. So how is nature going to select, oh, that's better than something else that's meaningless? Nature is not going to know what the difference is, and so there's no directed evolution in this case. So you can't use that argument. Yet he published that in his book, The Blind Watchmaker. That's as good as it gets, and yes, he says, I'm not really trying to illustrate how evolution really works in real life. This illustration is just to show you the power of natural selection. Well, I agree. If natural selection actually works, it's very powerful. But what happens when it doesn't work? It's not helpful. Specified complexity. The second major argument for intelligent design comes from William Dembski, who's a mathematician and philosopher who argues that nature is rife with examples of non-random patterns of information that he calls complex specified information. CSI for short. To qualify as CSI, the information must be both complex and specified. The letter A, for example, is specific. It's not like Z, it's specifically A, but it's not complex because it's only one letter. The string of random letters, such as Swiffler, on the other hand, is complex, sort of, but it's not necessarily specific. It's just a random jumble of letters. A Shakespearean sonnet, however, is both complex and specific. So, is Dembski's hypothesis falsified? Dembski claims that you cannot evolve specified complexity. So, Kenneth Miller, again, the same guy we saw before, he says, if Dembski were right, then a new gene with a new information conferring a brand new function on an organism can never come into existence without a designer, because a new function requires complex specified information. So, are there any examples of evolution producing complex specified information? And the answer, interestingly, to me anyway, is yes. There's lots of examples, hundreds of examples. And here's just two examples. The first example is nylon. Nylon was not invented until 1935. And there's a company producing nylon in Japan, and they took their byproducts of their nylon and they threw it in the back in this pond. And after a little while, they noticed that bacterial mats were growing on this pond, eating the nylon. And so, oh, wow, this is a brand new synthetic product. How in the world did these bacteria become able to eat the nylon and gain energy from it? And so they analyzed this bacteria, and they found out that the bacteria had a point mutation in their genetics that was a frame shift mutation. In other words, it wasn't reading. It's like taking typing on a typewriter and moving your hand over one place and typing again. What are the odds that that'll make any sense? So it's just about zero, right? And yet this is exactly what happened in the genetics of these bacteria. They had a frame shift mutation in their typewriter, and they typed a brand new GMOE sequence that happened to produce a nylonase enzyme to break down nylon, and it worked. It actually was functional. It broke down nylon, and it was about 300 amino acids long. And it worked. So that's one example, producing specified complexity, right? That's a fact, no argument. That's true. The next example is Barry Hall. He did experiments with lactase and E. coli. He deleted the lactase enzyme, which digests glucose, or galactose and glucose and fructose and glucose and lactose. And he deleted that enzyme, and then he grew those bacteria on lactose-enriched media in a lactose-enriched environment, and they evolved a brand new enzyme, an enzyme that did not have the lactase ability before, and with a single point mutation, it gained the lactase ability. And that's a brand new function. It wasn't there before. Random mutation, natural selection produced specified complexity. Correct? True. It's true. Aha, Dembski is wrong. Natural selection random mutation can produce specified complexity. Dembski is wrong, and I agree with that. Dembski is wrong in that aspect. The problem is that he's only a little bit wrong. Okay, what happens is if you look at the rest of the story, there's low-level examples that happen all the time. I talked about antibiotic resistance yesterday. That's almost always based on single point mutations, and you gain a new function. It's truly a new function. Sometimes it's a new enzymatic function that's not based on the destruction of something else. It's easy to break Humpty Dumpty, right? Most forms of antibiotic resistance are the result of breaking something that was already there working just fine. You break the interaction between antibiotic and target. That's easy to do because there's so many ways to break stuff. However, there are certain forms of antibiotic resistance that are not based on breaking something. They're based on the evolution of something brand new that wasn't there before, like penicillin. Penicillinase has never been observed to actually evolve in real time, but it may have evolved because it's a single protein enzyme, and we know of other specific enzymes that have evolved, like lactase and nylonase. So, and it's about the same size. So it may have evolved because it's at the same level of the other enzymes we know have evolved. But you go beyond the 300 to 400 amino acid level, and there are no examples beyond that. Also, if you delete the second enzyme that evolved in E. coli for lactase, it never evolves another one, another enzyme, another lactase enzyme at that level. So it's limited. There's limited evolutionary potential. Yes, there's evolution, and it does work, but it only works at very low levels, like 3-letter words for the English language system. The thing is that proteins are more flexible than English, and so that you can evolve more characters. Like, 300 or 400 letters in the English language system, that's a sizable phrase, a big paragraph. Whereas in the protein system, since they're more flexible in what can be substituted or changed, that's not so complex because it's not as high level of specificity that's required, as specificity in the English language system is on a higher level. But on a given level of specificity and size, you can only go so far before evolution stalls out at an exponential rate. And here you can actually see a visual illustration. These are real mappings, hyperdimensional projection on the three-dimensional space of proteins that we know about that are less than 370 amino acids in size. And you notice those that are 30 amino acids in size that are dark blue, they're closely clustered together in space, in protein space. Whereas as you go to larger and larger proteins, they get more and more scattered, more widely separated in space. And so, does my theory make sense? The higher level you go, the stepping stones get spread out? Yes, they do, and we know that they do because this is an actual projection of proteins we already know about in our dictionary. And here's another illustration, a schematic diagram of how there's a lot of space between known proteins, and these are just single protein systems. When you start moving up to multi-protein systems like flagellar motility systems that have 50 or so proteins, then it gets exponentially more spread out in space.
Is that we have
I'm sorry, this is the design flaw argument. Those who have to go, I don't mind. It's already 10 after. I'm a bit behind, but we've only got about 5 slides left. This is the intelligent design, or the flaw argument. Would an intelligent God make all these mistakes?
the implication of intelligent design is that God is a very, very bad engineer. I mean, an engineer who have designed an eye with the optic nerve having to cross the retina will be fired. An engineer that would have designed a human jaw would be fired. Our jaw is not big enough for all our teeth. God making this trivial, obvious mistake and errors of design, well, maybe their God does those things, certainly not mine. I don't want to have to worship a God that is not smart enough to do as well as a human engineer.
Okay, this guy's famous. Francisco Aya. He's a Don Brennan Professor of Biological Sciences, Department of Evolution Biology at UC Irvine. And he says, you know, what's God doing? He's making all these things backwards and upside down, and they don't work quite right. The jaw is too big, or teeth are too big for a jaw, and these are real problems, you know, for a lot of people. So how do you answer this? Well, I don't have time to deal with all of the design flaw arguments, but let's just deal with one of them that Richard Dawkins always talks about. Is the eye, the human eye is wired backwards. The wires in our eyes, the nerves, come in front of our visual field and then go back down the optic nerve into our brain. And so for light to come in through our eye, well, I'll just use his words because he describes it pretty well. Any engineer would naturally assume that the photocells would point towards the light with their wires heading backwards towards the brain. He would laugh at any suggestion that the photocells might point away from the light with their wires departing on the side nearest the light. Yet this is exactly what happens in all vertebrate retinas. Each photocell is, in effect, wired backward, with its wire sticking out on the side nearest the light. The wire has to travel over the surface of the retina to a point where it dives through a hole in the retina, the so-called blind spot, to join the optic nerve. This means that the light, instead of being granted an unrestricted passage to the photocells, has to pass through a forest of connecting wires, presumably suffering at least some attenuation and distortion, actually probably but not much, but still. It's the principle of the thing that would offend any tiny-minded engineer. Now, who would wire their photocells backward? Hello? So here's how verted retinas look, and actually some animals have this, like octopus. They have it wired the correct way. Insects, too. Some insects have it wired. But all vertebrate retinas are wired like this. Here's the wires coming in. Here's the path to the light. Light has to go right through the wires before it gets to the photocells to detect the light, and then it gets scattered a little bit. You know? And so who would do that wire it backwards? I mean, come on. And so, but there's some advantages. The advantages of the inverted retina, it allows for greater visual acuity and image processing power. The cost of visual acuity for humans, for example, is it costs a whole lot. The brain's cortex uses 300% less energy than the retina per gram. The renal cortex uses 50% less, and the heart uses 600% less energy. The choroid layer, this choroid layer right here, it has these vessels in it that supply the blood and nutrients and take away waste. It's in close contact with the ends of the photoreceptors right next door, right? It provides a large blood supply directly, really close to the photoreceptors because they use a lot of energy. It also processes large quantities of waste, approximately 100 segments per day per receptor. It rejuvenates the transretinal molecule to 11-cis-retinal, which is the molecule needed to actually sense the photon as it comes in, and then it conforms to a different shape, and then that signal is sent to the brain. It also provides a black pigment which prevents light scatter and reflection. None of these options are available to the octopus, which really doesn't need to see that well. In fact, if our retina, as it currently stands, was pointing the other direction, just the waste problem alone, these discs that are discarded every day, if they're discarded the other direction, away from the choroid layer, they could not be absorbed as effectively, and they would produce a hazy film in front of your visual field. That problem alone would prevent high visual acuity. Then, recently, just last year, May 2007, this was published in a very prominent science journal called PNAS. They discovered these Mueller cells, and you can see that these cells are elongated or widened on the light receiving end of the neural layer. These are the nerves going through the layer that the light has to go through. This is the source of light up here. These things catch the light here and fiber optically transmit it to the photoreceptors. And Dawkins didn't know about that when he wrote his book. These are just discovered last year. And so what little light scatter there might have been, it was taken care of by these Mueller cells. And those just happened. That's pretty much the end if you want to stay for question and answers, and I have a couple more interesting short clips that you may want to see. Otherwise, you're free to go. Sorry, let me get this sound down. This is the inner life of a cell that I think is very interesting. This is the blood vessel. These are macrophages traveling along the inner side of the blood vessel. They adhere to these molecules on the inner surface of the blood vessel. There's signals, areas of infection where the macrophages go to fight infection. This is a cytoskeleton on the outer surface of the cell that holds the cell together. This is the inner cytoskeleton that holds the inner side parts of the cell together. These are microfibers that are formed wherever they're needed, and when they get too long, they're modified by other proteins. They attach in just the right place and break apart the extra tubule that they don't need. This is a microtubule that's used for transport and also motility of the cell. It's broken down on the end, but it doesn't need to go on. This is a vesicle transport apparatus that actually walks along the microtubules to the places where this vesicle needs to go to provide nutrients and waste for the cell. This is a centromere that helps to divide the cell. It also has its own DNA. This is a nuclear pore that exports the messenger RNA into the surrounding cytoplasm. Then the ribosome we saw before comes on and it makes the messenger RNA very quickly. This is the mitochondria, the energy storehouse of the cell, the energy production of the cell. These are endoplasmic reticulophores where the protein gets deposited for further modification. More walking. This is the Golgi apparatus, where the proteins get further modified before they're exported outside the cell or to other places within the cell. And then they get transported with that little walking thing. And then they get extruded, exocytosis, in the cell membrane. When this little circle gets big enough, it reaches a certain threshold and it stimulates these proteins to unfold and connect with cells on the other cells outside of the cell with their protein sensors. There's the macrophage again. It's going to come to the right spot with chemical signals and go through the cell wall, or the vascular wall, to go attack the foreign invaders at just the right place. Anyway, just think that's quite amazing to me. And like you said, if you know enough about it, yes, evolution happens, but it's almost trivial, the amount of evolution that actually happens, because nothing close to that, or even much lower level systems, evolution doesn't come close. And statistically, if you sit down and look at the math, it can't come close in trillions and trillions of years. That should have 4 or 5 billion years, even according to mainstream thinking. So intelligent design, I think, is at least a reasonable option, if not obvious. So, any questions? Yes?
The process of protein production actually requires protein.
Yes.
You kind of have a chicken and egg problem there. It seems like a simple argument for evolution. I remember when I was in high school biology, that was kind of an argument, a simple argument that made this evolution. Has that been addressed? It seems so obvious, this idea of chicken and egg. Which came first, the ribosomes and codons, or the DNA, the RNA in translation? You have to have both.
Right. Yeah, I present that argument on my website, and interestingly enough, if anybody's seen the Ben Stein movie, No Intelligence Allowed, or Expelled No Intelligence Allowed, he asked that question of several of the leading scientists of the day, and most of them say that no one knows the answer to that. Some of them say that they think they know how it might be an answer, which is that it started out with messenger RNA, or an RNA molecule, or self-replicating proteins that did the job before the need to have the whole system in place. But then when you ask them for more details about how that might happen, or maybe if there might be an experiment to demonstrate at least the early steps in that process, well, they don't have that. And so, ultimately speaking, they say, well, nature did it somehow, we just don't know yet. Someday we will know, but we know that nature did it. The same thing like us, we know that God did it. But yeah, that argument has not been satisfactorily addressed in the scientific community, and they admit it, most of them. Even Richard Dawkins admits that one. So what I usually do is I say, okay, that's abiogenesis. That's around before natural selection is in play. And I was like, okay, we won't argue about that. Especially with theistic evolutionists. A lot of people believe that God started it because, oh yes, the first cell, that's way too complicated. But after the first cell, then Darwinism is in play, random mutation, natural selection. Then we can have evolution. And so I was like, okay, forget abiogenesis. We'll give you the first cell. Let's just go from there. And does evolution actually work from there on out? And I was like, okay, it does sort of, trivially, at very low levels. But nothing that would give you any sort of unique systems that are very much beyond what you started with. Anybody else? Yes.
I've heard that basically they've gotten rid of the idea of evolution of the first cell even occurring on Earth, that it came on a meteorite or something of this fashion, and so therefore they're arguing that point away from the creation of the first cell.
Yeah, Sir Francis Crick, actually, one of the men who came up with the double helix for DNA, he actually presented evolution from outer space.
Exactly.
He says that, yes, the odds of evolution happening, of the first cell happening here on Earth are so remote that it's impossible. So it must have been planted here by aliens or brought here by a meteorite or something from outer space, because outer space is so big, there's so much room there that it must have happened somewhere else and then brought here. But you know, how many, we kind of have a, according to mainstream science, how many atoms are there in the whole universe? There's only, anybody know? There's 10 to the 80th. 10 to the 80th. And let's say the entire universe was filled. We also know the dimensions, the diameter of the known universe. Let's say all of that was filled with bacteria. How long would that significantly reduce the time needed to evolve a flagella? Statistically. You know, would it change the odds? He's saying the odds are too great, or the first single cell. The odds are too great just on Earth. But given the entire universe with all the atoms, in fact, even more than all the atoms, fill the whole universe with atoms, all bounced together randomly, everything that you need, make it the whole universe open to that. And we'd still have the same problem. Statistically, it only changes it by a few orders of magnitude, about 100 orders of magnitude, whereas the orders of magnitude that convinced Crick that it couldn't happen here are in the thousands. That's the problem. It doesn't really solve the problem, even given the whole universe.
Back to simpler questions. Why does the mitochondria have DNA anyway? And why is it only female DNA anyway? Why is it, what does the mitochondria do with DNA back to the cell?
Okay, the question is, why does the mitochondria have its own DNA, and why is it only female? Why is it only inherited maternally? It is only inherited, well, let's start with that question first. It's only inherited maternally because the spermatozoa doesn't have enough, when it deposits its genetic material, its mitochondria get left outside of the cell, outside of the female egg. And so when the female egg starts dividing and producing the embryo, the only mitochondria that it has access to are the ones that are originally inside the female cell. And so that's why there's only maternal inheritance, which isn't actually completely true. Lately, in the last few years, it's been discovered that sometimes male mitochondria get inside that cell, and I mean, they just happen to be aggressive, I don't know, and charge extra hard, and get inside it somehow. And so sometimes there's maternal influence on mitochondria as well. But, yes, sir?
Well, what about the devil as intelligent design? He had something to do with dinosaurs and all those things, and the condition of the earth before the flood was entirely different too. That's another factor.
Yeah, there's several arguments based on the devil's ability to design, and I think he is more capable than we are of design, of intelligent design. A lot of people say, oh, the devil can't create. That's nonsense in my view. I was like, if we can create, the devil can create even more. And so yes, I think the devil could have had a play in design elements, but that's still design, in my view. Some people also take that farther and says, well, the devil may have manipulated the planet for millions of years, and that's why we have these old fossils and all these failed experiments of the fossil record. And we can go further into that probably more tomorrow, because I talk about fossils and stuff tomorrow in geology. Personally, I don't buy that argument for several reasons, and I can get into that further. Probably tomorrow would be a better time for that. Yes, sir.
Evidently, is plenty of evidence scientific that we can create. What are they?
That we can create? Well, have you ever made anything like a pie? Did you create pie? Right? So we can create, at least on a certain level. What about cars? Right, but I mean, there's certain things we can't.
I'm thinking of life.
Right, well, it all depends on your definition for life, too. Like here, Francis Collins, the guy who first sequenced the human genome, he says in the next couple of months they're going to make life. Well, it all depends on what your definition of life is. But we can make pretty complicated stuff. And so if you want to call that life, it's all a definitional thing in my book. Yes, sir?
Do you mind explaining a little bit more that three-dimensional scatter plot of the amino acids?
Yeah. At first I didn't believe it. Somebody was telling me that protein sequence space and genetic sequence space was hyperdimensional. It's more than 3 dimensions. And it's actually, the number of dimensions has to do with the size of the sequence. For example, if you have a protein that's 20 amino acids long, it occupies 20 dimensions of space. And for me, I can't picture that in my mind, but mathematically, the numbers, it's true. And to explain it up here without using a paper or using the statistics for you is difficult. But statistically, the sequence space occupied by genetic sequences is hyperdimensional. It involves whatever size of the sequence you're talking about. And I can sit with you personally later and explain that to you. And I didn't believe it until I actually worked out the numbers, and it's true. Anybody else?
Can you tell the part that location plays the gene that is located?
Yeah, the location of a gene or genetic element or something that's actually functional within the genetic sequence is actually important. If you, like say, for example, what's the difference between a horse and a donkey? They can produce a horse and a mule. They're all the same gene pool. Same thing with a horse and a zebra. It's just the only difference between them, all these creatures that share the same gene pool, is the location of the genetic sequences on the chromosomes. They have chunks of chromosomes that are in different places, and that's what makes them look different. Otherwise, they have the same genetic elements. It's just that you take a certain genetic element, you put it in a different place in the chromosome, you may get more expression or less expression of that genetic element and change the look of the creature. Anybody else? Thank you all for coming. Appreciate your time.